Just to check, you see the correct slide, correct view. Great. So maybe I'll then go on. Welcome everyone for this monthly seminar that's organized by the Network for the Integration of Speciation Research. And this is a ESAP-funded special topic network, and our aims are to promote a framework for integrative speciation research and to promote integration of speciation researchers, diversity and inclusiveness in our field. And the seminar series will be 1 hour of talks. And after that, there's a general discussion about 30 minutes. The talks will be recorded and provided later, but the general discussion will not. And there is 1 seminar per month, and the time slot alternates so that people from different time zones can join the seminar. And since this is a webinar format, please post your questions and discussion points to the chat. But before we begin the seminar, I would like to introduce or announce a workshop that the network is organizing. And the theme is how we can better integrate speciation research. And this workshop will be held in Tvärminne Field Station in Finland at the end of March next year. And we are hoping to gather a diversity of career stages, perspectives, and backgrounds to this workshop. And with this diverse group, we would like to discuss how we can bridge existing gaps in speciation research. And the network has identified some priority areas for discussion and invited discussion leaders to lead discussion in these topics and working in small groups. And the outcome of this workshop is planned to be a perspective paper, for example, in evolutionary biology or another of the society journals. So please keep your eyes open. Please apply, and more information will follow in our mailing list and Twitter. And now on to Roger and Richard.
Okay, so it's my great pleasure to be able to introduce our first speaker, who is Professor Katie Peichel from the University of Bern in Switzerland. I think Katie doesn't need much of an introduction for those of us who work on speciation. So amongst other things, she's organising this called Cold Spring Harbour special issue on speciation, and that a lot of us are involved with, and also the Gordon Conference on speciation, which is coming up in January. But I'll briefly describe Katie's very, very impressive career. So following her undergraduate studies at Berkeley, she then studied for a PhD at Princeton, supervised by Thomas Volz, and I believe working on mouse developmental genetics, which is slightly different from what she's doing now in some respects. But after this, she started working on sticklebacks at Stanford with David Kingsley lab. And I think it's the stickleback work that we really know in this community, Katie, for now. As president, after a brief stint as a professor in Kathmandu, she then started her own lab at the Fred Hutchinson Cancer Research Center, focusing not on cancer, obviously, but on the genetics of complex traits. And then more recently, in 2016, she moved to Europe, which is now professor and head of the Evolutionary Ecology Division at the University of Bern. So she's won a number of awards, but was most recently elected a member of the American Academy of Arts and Sciences. And I think this is especially surprising if you know Katie's work. So she and her lab continue to publish really exceptionally high quality papers on a number of questions, including, for example, how and why do sex chromosomes evolve, the genetic and neural basis of behavioral evolution, and importantly for us, of course, the genetic basis and genomics of adaptation and speciation. So thank you for agreeing to talk to us today, Katie, and I'm really looking forward to your talk.
Well, thanks, Richard, for that overly generous introduction. I hope I can live up to that. It's really an honor to have the opportunity to talk to you guys today. A little bit on my thoughts on what QTL mapping can tell us about the genetic architecture of speciation. And so I want to start maybe a little bit controversially about and just providing you with the species concept that I'm going to use as a framework for my talk today. So the biological species concept as put forward by Mayer suggests that reproductive isolation is sort of the key to understanding speciation. This definition was sort of updated by Coyne and Orr, suggesting that that we could still define things as species even if they weren't completely reproductively isolated from each other. And so what's crucial to the species concept is that the study of speciation is essentially the study of reproductive isolation. Now, I know this, not all of us agree on that. And even I sort of am coming to as you'll see in the upcoming GRC that many of you are going to attend, I think we also need to be focusing on sort of species persistence and not just reproductive isolation. But for the purposes of today's talk, I think this is a useful species concept. This is this nice paper by Sean and Mark Ravenet showing sort of what people use as species concepts just depending on different factors. But here for the field of genetics and genomics, one of these biological species concepts, BSC 1 or 2, or sort of the framework. So that's the framework I'm going to use for today. So for thinking about reproductive isolation, which again is a concept that maybe we don't all agree on, but I think we can all agree that there's sort of barriers to reproduction. And so we can think about barriers that occur before zygotes are formed, and then these barriers that can occur sort of once zygotes are formed, either intrinsic things like sterility and viability, or ecologically dependent forms of isolation. Okay, so I would sort of put forward the argument is that if we're thinking about the genomic architecture of speciation, we might be thinking about this in the framework of the genomic architecture of reproductive isolation. And I would sort of make that even more clear and say we're sort of interested in the genomic architecture of isolating barriers, or at least that's sort of the framework in which I think about the genetics and genomics of speciation. And the specific questions that many of us have been interested in for a long time, of course, are how many loci contribute to these isolating barriers? Do they have large effects on those barriers or small effects? How are they distributed across the genome? Are they sort of found wherever in the genome? Are they clustered in things like inversions or on sex chromosomes? And then for some of us, we actually want to get down to knowing what the molecular identity of the loci actually is. Okay, so how do we sort of go about doing this? The classical approach that's been used for a long time is a so-called forward genetic mapping approach, or this is also referred to as quantitative trait locus or QTL mapping. And the idea is that you basically start with species or populations that are experiencing some degree of reproductive isolation. And then we know what the isolating barriers are. So in the case of these fish, it may be that there's some form of hybrid sterility. There are differences in female preference or in male traits. And because, of course, this requires the ability to actually cross things. And so we cross these species that have some barriers and then generate many hybrid offspring. We can score those offspring for these barriers of interest, and then use genetic mapping to basically identify regions of the genome where we see these associations between the genotype and the phenotype. And if we're really, sort of motivated to go after figuring out what the genes are, going down to, okay, what are the genes in these regions, and then the actual molecular changes that cause these isolating barriers. And so, again, this has been sort of traditionally used. And around the turn of the last century, I would say our knowledge of the genetic and genomic architecture of isolating barriers was relatively limited to studies of traits like hybrid sterility and inviability, a lot of work in Drosophila, some work in house mice. And then there's, around, the early 2000s, there was also some work on things like sexual isolation, this really amazing pioneering work of Toby Bradshaw and Doug Shemsky on the Mimulus system, work on heliconia, some of which was done by Richard, work by Sarah Vaya on looking at habitat isolation. But yeah, sort of not a lot when we sort of think back like 20 years ago. And there's been some work from these QTL studies over the past couple decades to sort of get at the genetic architecture by QTL mapping of some pre-zygotic barriers and some post-zygotic barriers. But I'm actually looking forward, Richard mentioned this Cold Spring Harbor volume on speciation. I think there's going to be some really nice comprehensive reviews on this topic, which I'm not involved in, but I'm really looking forward to seeing those. But I wanted to talk a little bit just about the sort of the QTL mapping approach in general and sort of the advantages and disadvantages. So, what I think is the strength of this approach is that you're really starting with isolating barriers that are relevant for maintaining sort of reproductive isolation. So you're starting with phenotypes of interest. With QTL mapping, you can sort of actually get estimates of how large an effect those loci have on these barriers. You can look at the distribution of those loci in the genome. Are they clustered or not? And this is a starting point to sort of figure out what the molecular basis of that barrier is, if that's where you want to go. The disadvantages, of course, are that it's a lot of work to figure out what are the relevant isolating barriers in nature. And James Sobel, in one of the earlier talks in this series, really emphasized, I think, the crucial importance of this. And I really agree with that. It's hard to do, but it's important to do. Of course, this approach is limited to species that you can either hybridize in the lab or that can hybridize in the wild for things like admixture mapping or in hybrid zones. You still don't know if you see clustering. QTL mapping sort of gives you broad regions of the genome that are associated with traits. It could be that there's a single pleiotropic mutation. It could be that there's multiple linked mutations. So you're sort of limited in what you can infer about what forms those clusters. And then, if you want to go after the genes, the QTL are large and they contain many genes. So the path to identify the molecular basis is long. So, QTL mapping is sort of hard to some and limited to some extent. We can't do this for all species. So in the past 10 to 15 years, people, were really excited by the idea that next generation sequencing could give us some insights into the genomic architecture of speciation. And so what we hoped, I think, is that we could go out into nature, we could take any two species that are reproductively isolated and sequence their genomes, and that was going to tell us sort of what the traits and the genes were that were relevant for reproductive isolation. So what we hope to find is, I think what Leo's going to show us a beautiful example of in the next talk, is we sequence a genome of these two birds, for example, and we see these really clear, beautiful peaks of divergence, and there's, really great candidate genes in the middle of these peaks. And so we have some insight into speciation just from sequencing the genomes, of course, combined with natural history, et cetera. But I think more often what we find is something that looks like this. This is just one early example from the flycatcher work showing sort of there's lots of peaks of divergence between these species. We started to realize that maybe these peaks are found in regions of low recombination. And so sort of what are the sort of loci here that contribute to barriers between the species is not entirely clear just from looking at the comparison of these genomes. So the speciation genomic or population genomic approach, again, sort of has advantages that can be applied to any system that you can get DNA from. You could sort of identify whether loci sort of are highly divergent or not, but that doesn't necessarily tell you if it has large effect on reproductive isolation. We can look at clustering, and we can sometimes pinpoint real candidate genes within these regions. Disadvantages are, I think this is the biggest one that many of the people here have worked on, is like, what are the targets of selection? What are just the effects of being in a region of low recombination? How do we disentangle recombination, background selection, et cetera? It can be challenging. I think for me, the challenge is, so just because a locus it looks like it diverges between species, is what isolating barrier does that actually control? And so that, I think, is a real challenge, is linking these loci to specific phenotypes and specific isolating barriers. Again, same as with QTL mapping, we don't necessarily know if we see like a big inversion, is that due to a set of linked mutations or pleiotropic mutations. So I think the ideal approach is really to combine these two approaches, forward genetic approaches and sort of the speciation genomics approaches in systems where that's useful. But I think, again, I want to really echo this point that James Sobel made a couple lectures ago, is that the key here in both cases is really knowing your system, understanding the ecology and the biology of your system, including what the possible isolating barriers are. And that really gives you the ability to sort of interpret genome scans or sort of direct your QTL mapping to traits that are really of interest. So that's sort of one takeaway that I'd like everyone to think about, is that this deep knowledge of a system is really crucial, I think, no matter what approach you're using to study speciation. Okay, so I just want to maybe hopefully have a little bit of time to just say what I think we've learned from QTL mapping in at least one system, the system that I work on, about these questions. And so today I want to focus on these benthic limnetic species pairs, which are found in a few lakes on Vancouver Island or near Vancouver Island in British Columbia. And within these lakes, there is a large deep-bodied benthic species that lives in the nearshore habitat and is feeding on macroinvertebrates. And there's a second limnetic species that's living in the open water and is feeding on zooplankton. And there's all these morphological differences between benthics and limnetics that we think are key to sort of feeding on these different resources and living in these different habitats within the same lake. Now, from a speciation genomics perspective, if you sequence the genomes of, in this case, 3 of these benthic limnetic pairs, you see these patterns that are very repeatable across the 3 independent pairs. But basically, you see this really high divergence in the middle of chromosomes and low divergence sort of at the ends of chromosomes, consistent with these being in the regions of low recombination. So again, we could sort of look at this pattern and just say, well, which loci are important here? I mean, lots of the genome seems to be under divergent selection between benthics and limnetics. How do we tie these to phenotypes? And so, but luckily in this system, really due to pioneering work by Don McPhail and then Dolph Schluter and their colleagues and students, we know a lot about the isolating barriers between these benthics and limnetics. And so we know that there's strong assortative mating. So there's strong female mate choice and divergence in male traits. Where there's also this habitat, so there's ecological isolation based on food resources, but there's also differences in, say, for example, where the males put their nests. There's not a lot of temporal isolation or gametic isolation, and then there's really no intrinsic barriers. So there's no hybrid sterility or hybrid inviability in the lab. But there is ecologically dependent hybrid inviability. So what I'll call here extrinsic incompatibility. So hybrids are formed in the wild to some extent, but then there is strong selection against them. Okay, so we use this knowledge to try and go after the genomic architecture of these isolating barriers. And this was a project that Dolph and I started quite some time ago, and we were quite lucky to get funding from the NIH to do this. And we were also very lucky to work with a really talented postdoc, Matt Arnegard, and graduate student, Gina Conti. And this work was done when I was in Seattle, so I wasn't so far from Dolph's amazing experimental ponds at UBC. And so these ponds basically contain a deep open water zone that allows vertical migration of zooplankton, and this more shallow littoral zone, which is a more benthic-like habitat. And we can really recapitulate the natural habitats in these ponds. So you can find benthic males nesting in these beds of chara, and limnetic males nesting in these sort of more open habitats, just as they do in the wild. And so what we did is we crossed benthics and limnetics for this QTL mapping approach. We made F2 hybrids. And then the goal was to phenotype those fish in these ponds for all of the sort of isolating barriers that we know exist, genotype, and then do QTL mapping. And this first set of data is looking at sort of morphology and hybrid performance, or sort of this extrinsic reproductive isolation. We had 633 of those F2s, and we genotyped them with SNPs. And this work was published quite some time ago, so I'll sort of just quickly go through it. So the first point is that when we map the sort of what the fish are eating based on stable isotope signatures, this is all 633 F2s in the ponds. And then we use body size as sort of a measure of performance. And so we see these sort of peaks of body size here and here, and then this valley of very small fish. And when we look at what those fish are eating, we find that these B fish are eating benthic resources. These L fish over here, which are also large, are eating zooplankton. These weird A fish are eating sort of a weird resource, and then they're sort of all eating cladocerans. Okay, so this major axis of variation here, this principal component 1, we're calling niche score. So it really is differentiating the benthic and limnetic feeding fish in these F2s. And this sort of niche score is associated with variation in traits that we know are important for feeding. So for example, the L fish have limnetic-like gill rakers, suction feeding, and these sort of jaw traits, and the benthic-like fish have benthic-like traits there. So I'm just highlighting some of these key traits, but it's not that simple. Basically, over half of the traits we looked at were associated with variation in niche score. So all of these traits are sort of contributing to how the fish are sort of performing along this benthic limnetic axis. And when we map the genetic basis of that, we find that they basically map all over the genome. We do find some clusters on 16, 20, and 21, but it's really a polygenic genetic architecture of these traits. And then when we look, sort of try and map genotypes back onto this niche score, we find that there's 11 of these genomic regions that contribute to variation along PC1. And they're acting additively. So basically, it's just the number of benthic alleles that you have at these 11 loci. Sort of, if you have few benthic alleles, you're more limnetic-like, you're over here. And if you have more benthic alleles, you're over here. So it's really this polygenic additive basis for movement along this niche. So just briefly, what about this PC2, which is really these weird, very small A fish? So these fish actually have a phenotypic mismatch. So in this in their lower jaws, they're very limnetic-like, and in their upper jaws, they're very benthic-like. And so this is this oral jaw trait. So the idea is that they basically just have these messed up jaws that don't really allow them, that don't work well together and allow them to feed on normal food resources. And they were feeding on sort of this weird resource in the ponds. And when we look at the genetic basis of that, we see that there's a strong QTL for this lower jaw trait, which is unlinked from this QTL for upper jaw trait. And we see sort of maybe some evidence for epistasis and interaction between them. And I think this is very similar to the sort of intrinsic incompatibilities that we see, where we see these sort of epistatic interactions between loci. And actually, Ken Thompson, who I think is here today, has sort of looked at this at a genome-wide level. So in this very clever approach, he sort of compared, so under this model, you would expect that you would have fish that are sort of LL genotype at one locus and BB genotype at the other are just not doing well. You have these incompatibilities. But in the lab, there's no intrinsic problems. You would see these genotypes showing up. And so overall heterozygosity should be sort of what you would expect under Mendelian ratios. But if there's extrinsic isolation, you would sort of expect an excess of heterozygotes because these homozygotes are basically being selected out. And indeed, when Ken compares genotype data from fish benthic limnetic F2s raised in the aquaria and those raised in ponds, we basically see this excess heterozygosity, suggesting that these sort of extrinsic incompatibilities are actually found genome-wide. Okay, and so I don't want to take too much time now, but we also looked at the genetics of female mate choice. I'm not going to run through those results. I'm happy to chat about them, but just basically get to the punchline here, which is the genomic architecture that we've seen from these QTL studies is that there's many loci, and they're mostly additive of small effect, sort of distributed across the genome with a little bit of clustering. Of course, we still don't know the identity of these loci. And I would say the implications of what we've learned from these QTL studies are that even when you have species evolving in the presence of gene flow, you can get this distributed genetic architecture that contributes to reproductive isolation. I, a priori, really thought we were going to find a simple genetic basis here because of this, because these fish are living in the same lake and they do hybridize. But maybe the presence of sort of strong multifarious selection is enough to overcome this. We also are probably dealing with a situation where we have selection on old standing genetic variants. But I think what's cool here, and what I didn't really have time to show, is sort of the traits that are important for ecological isolation are also important for mate choice. And these sort of called magic traits don't need to have a simple genetic architecture. That sort of reproductive isolation emerges, or assortative mating sort of emerges, even when you have this sort of polygenic architecture. And also, I think it's really cool to think about the similarities between the genetic basis of extrinsic postzygotic isolation, so this phenotypic mismatch, with genotypic mismatch that we think of from Dobzhansky-Muller incompatibilities. Okay, so I will stop there and hope I haven't gone too far over. And if there's time for questions, I can take them. Otherwise, I'll save them for the discussion.
Great. Thanks very much, Katie. That was a really wonderful talk. I think we have time for one or two very quick specific questions for Katie. If anybody wants to post in the chat.
Roger has a question.
I'll let you just ask it, Roger.
Yeah, if nobody's going to post anything in the chat, I'll ask. I just wondered if you could comment on the difference between the QTLs for barrier traits and the QTLs for the actual barrier effect. So often what you're measuring is the genomic basis of some phenotype like preference or physiology, and not actually the barrier effect, which may be quite difficult to infer from the phenotypic effect size.
That's right, Roger. It's true that our effect sizes are for the phenotype, the isolating barrier, but then you would have to do some calculation to say, okay, well, what is, if we know the contribution of that barrier to overall reproductive isolation, then we can have an estimate. So in some systems I worked on in sticklebacks, we do have that, like the Japan C system. So we can sort of say, okay, overall this trait contributes this much to isolation, and this QTL explains this much of the variation in that. So but you need to know the contribution of that isolating barrier, of course, to overall
and more of the biology, so how the preference relates to the variation in the male trait or whatever. Yeah,
Exactly. Yeah.
Yeah, thanks.
Mhm.
I think we should probably move on in the interest of time, but there'll be plenty of time for more general discussions after Leo's talk.
Yeah, great. So yes, it's my great pleasure to introduce Leo Campahna. Leo did his PhD at the University of Buenos Aires, and with also an association with Queen's University in Canada, and then moved to a postdoctoral fellowship at Queen's University. And from there to a position at Ithaca in the Lovett Lab. But from 2016, he's been a staff member at the Cornell Lab of Ornithology. He uses genomic tools to study the evolution of avian systems, particularly diversification and particularly early stages of speciation. And so he's done some really fascinating work on these South American finches, which I hope we're going to hear more about in a minute, particularly focused on the role of sexual selection and sexual signaling, both plumage and song, and its contribution to isolation, trying to understand processes, not just genomic patterns. But interestingly, Leo also works with more comparative approaches, using, for example, demographic inference to look at the impact of barriers on diversification in South American birds more generally. Of course, that combination of looking at directly at mechanisms and more comparatively is very close to our hearts in the integration network. It's just the sort of connection we're interested in making. So we're very much looking forward to hearing your talk about prezygotic isolation and the genomics of avian plumage coloration. So take it away.
All right, thank you, Roger. Let me see if I can share. There we go. All right, everybody, you guys can see that well, right?
Perfect. Yes, thanks.
Excellent. Well, thank you very much. Thanks for the invitation. It's really a pleasure to be able to present here. And thank you, Katie, for a really good introduction that sets me up for what I want to talk about today, which is the genomic architecture of avian plumage coloration. And I want to talk about this in the context of two systems where we think it's particularly relevant for generating or contributing to generate prezygotic isolation. And I wanted to start by putting up some tables and figures from classic papers that have shaped the way I think about this. And first, I wanted to start with this classic paper by Peter and Rosemary Grant from 1992. I remember I read this probably in my first week of grad school, and I was coming from immunology, from human immunology, so this was news to me. To see here, this table, how they break down the proportion of species that are known to hybridize across the different avian orders. And we can see a lot of variation. For example, ducks and geese do something different from the rest of birds, but on average, the conclusion, the punchline is that about a little bit over 9% of species, of avian species, are known to hybridize. The next, I wanted to show this graph from a paper by Price and Bouvier from 2002. And on the x-axis, we can see sequence divergence between pairs of species that hybridize, and so as a proxy for the time they've been diverging. And on the y, we see a fertility index. And of course, you know, the higher up you are there, the more, the higher the fitness consequence that these hybrids are paying, or the species that are hybridizing. And so, of course, as species get more deeply diverged, we tend to observe more fitness consequences. However, I wanted to sort of point out this quote that I pulled down in the bottom that says that species that produce infertile hybrids last shared a common ancestor on average about 7 million years ago. And the third piece of the puzzle here is from Jason Weir and Dolph Schluter, where they're showing the age between sister species pairs of neotropical birds. And the point is that there is a latitudinal signal, but the reason I'm pointing it out or bringing it up here is to show that on average, these sister pairs are way younger than the 7 million years that I pointed to, that I referred to in the previous slide. And so, sort of to summarize, we know that many bird species can hybridize, and that on average, only relatively old pairs produce infertile hybrids. And that many sister pairs, or most of the sister pairs that I was showing just now, are younger or relatively young. So the question I want to talk about today is the evolution of prezygotic reproductive isolation mechanisms particularly important in avian speciation? And I wanted to talk about this in the context of these two systems. So first, I'm going to talk about the independent origins of melanism in the chestnut-bellied monarch from the Solomon Islands. And then I'll end talking about cappuccino seed eaters and the evolution of plumage patterning in these South American birds. And so the work in the chestnut-bellied monarch is done in collaboration with my friend Al Uy, who's worked on this system for a longer time. And Al has shown that on the main island of Makira here in the Solomon Islands, birds have a chestnut belly. And so then there are two groups of satellite islands, Ugi to the north and Santa Ana and Santa Catalina to the east, where birds are completely melanic. And if we look at this tree on the right, you'll see that these two melanic populations are not each other's closest relatives. So we think that melanism has likely originated twice. And so using a candidate gene approach, Al's been able to show that the birds on Ugi carry a derived mutation on the agouti signaling protein, while the birds on Santa Ana and Santa Catalina carry a derived mutation on MC1R. And of course, this receptor ligand pair controls sort of the ratio of eumelanin to pheomelanin, so black pigment to brownish pigment. And we can see that there's three different phenotypes that we can find. So as I referred to before, the chestnut-bellied birds, they all carry the ancestral variants, and they're homozygotes for that in Makira. The melanic birds, the fully melanic birds, can carry the derived mutation either in ACIP or MC1R, depending on the population. And we find a low number of heterozygotes that have this intermediate phenotype where most of their belly is melanic, but they have a little patch that is brown. And so this trait, this coloration trait, seems to be relevant for species recognition. And in these experiments, these are simulated territorial intrusions. So Al's found a territorial male and exposed them to combinations of song and plumage traits in these simulated intrusions to see how the bird would react. And we see that birds react more strongly to their local plumage and song. So in Santa Ana and Santa Catalina, the strongest aggressive response is to melanic plumage and the song from this subspecies, and the opposite is true in Makira. Well, the same is true, but with the opposite phenotype. And so what we think is going on here is that the birds are using these traits for species recognition. At least we know directly that males are, and we think that females are probably using the same traits for mate choice. And what I want to ask in the next few slides directly is, what are the molecular targets and the evolutionary forces that has shaped this conversion evolution of melanism? And then ask, how has selection shaped this trait, which we think contributes to generating reproductive isolation? And so from now on, I'll show whole genome sequencing data. This is from a paper that came out in PLOS Genetics last week. If you're interested in the details, it's there. First, in the PCA, we can show that obviously individuals cluster by island, irrespective of their coloration phenotype. Our tree here in the middle, again, shows that melanic individuals are not each other's closest relatives. They're derived from Makira twice. And from demographic reconstructions, we see that actually Santa Ana and Santa Catalina is a much younger population than the population in Ugi. That's split from Makira about 8 times. The split is about 8 times older. And so next we used these different phenotypes, individuals from these different phenotypes in the context of a genome-wide association study to ask what the genetic basis of this coloration trait was. In this case, we're comparing Makira individuals to those from Santa Ana and Santa Catalina. and we see that there is a single association peak right here on chromosome 11. We can zoom in to that peak right here, and indeed this is, as we expected, the region that contains MC1R, including that coding mutation that I had talked about before in the context of the candidate gene study. We can do the same for the UGI population, and in this case, remember this population is a little older, so there are a few more divergent peaks. These different peaks in total contain about 83 annotated genes, but ACIP is the only coloration gene that we know of that is found here on chromosome 20. And if we zoom in, we do see that coding mutation on ACIP and other mutations that are associated to this color difference in the vicinity of the gene. And so next we wanted to understand how or what processes has shaped this region. And for this, we use this statistical framework here, in which we first take our inferred demographic model, and then we simulate different situations. We simulate neutral mutations, or we simulate beneficial mutations, and there are very many parameters. And so here's just a cartoon showing you, we'll see the frequency of neutral mutations in yellow as the simulation goes on. Here in the hard sweep situation, we've had in the center a beneficial mutation that rises to fixation and eventually will fix. And in the bottom, we have a soft sweep situation in which we've allowed a mutation to be neutral for a certain amount of time, and then we turn on selection and allow the simulation to progress. And so these are the patterns we're left with after these particular conditions of the simulation. We do this millions of times, varying the strength of selection, the time of onset of selection, and things like that. Next, what we do is we take those simulations and we infer ancestral recombination graphs. So what we're going to do is try to infer the most likely topology at these different variable sites, including in the center here our beneficial mutation. And the benefit here is that these are the most likely topology for different sites. So they're taken into account likely recombination. After that, we will extract features from each one of those topologies. For example, the number of lineages that remain containing either a derived or an ancestral mutation at different times, and use that to train a machine learning algorithm to recognize and distinguish these different simulated scenarios that we started with. Once we have that algorithm, we can apply it to our real data. And so here I'm showing the region that contains the MC1R gene. And on the Y, we can see the probability of a mutation not being neutral. The little triangles show our GWAS hits, and you can see that most of our GWAS hits are inferred to have undergone a soft sweep in this case. This includes the coding mutation on MC1R, and also many other mutations that sit on that same haplotype and so are inherited together. Here I'm looking specifically at that coding mutation, and we can see different characteristics that the model is inferring. So this is a rather older mutation. The onset of selection is actually quite recent, estimated at about 500 generations. It's estimated to be most likely a soft sweep, and selection is about 0.02, the selection coefficient. And so here on the top in blue, you can see the trajectory of the derived allele and how when we turn on selection right here, it rises in frequency really fast. And so just to compare these different parameters to our genome-wide inferences, we can see that the selection coefficient is actually one of the stronger ones that we observed in the entire genome. And the onset of selection is relatively on the younger side. So we can do this, the same thing for the ACIP region in the UGI population. And in this case, we actually find that most of our GWAS hits are inferred to be neutral. We do find some areas that have a high chance of not being neutral. And here's, for example, one that we think underwent a soft sweep. However, what we think is happening here is that we're applying this model to only the UGI population. And in this case, these GWAS hits are not showing any variation within UGI, so they're fixed. What we think has happened here is that we have a case of an older sweep that's already completed. And I think this also illustrates that these inferences are hard to make, even for a situation in which we have a relatively, what we think is a simple genetic basis, and the divergence is not that old. But there's the sweep seems to have completed already. And so here, just to finish this example, I'm pulling out topologies from these gene regions, taking into account all the populations just to show you how they look. In this case, this is a position on MC1R, and you can see that we have a clade here enriched for the blue population, which is the Santa Ana and Santa Catalina population. And we see the same thing in ACIP for the UGI population. And so far I've shown that we think that independent and very simple genetic changes are associated to melanism in the system. That selection has shaped melanism both through MC1R and ACIP on the different populations. In particular, in MC1R, we observe pretty strong selection that's relatively recent and has happened on what we think standing genetic variation. And selection on ACIP is perhaps relatively older. And so the other example I wanted to talk about were cappuccino seed eaters. And these are males in adult reproductive plumage of all 10 species of cappuccinos. You can see that they vary in different patches across their bodies. They vary in color. You can have, for example, a white throat here in Palustris, a black throat in Ruficollis, the cap, the belly. Certain fixed patches tend to change across different species. And always in the same sort of tones of brown, white, gray, or black. And so what we think happened here is that there's been rearrangement of existing genetic variation that has led to this evolution of plumage patterning. And this has in turn promoted reproductive isolation and the beginning of speciation in the seed eaters. And just so for a little bit of context, this group is interesting in that it shows very, very, very shallow genetic divergence. Basically, the tree for the group is a big polytomy, and you reach the same result using different types of genetic markers, some more classic, but then even reduced representation genomic techniques show you the same pattern. They are continentally distributed and highly sympatric. We can date the origin of the radiation to about 1 million years ago in the Pleistocene. And you can find them basically in the same regions, nesting in the same regions in Southern South America. And they're also, inhabit a relatively simple habitat, which are grasslands. So we don't see any segregation in the places that they're nesting. They are found syntopically nesting in the same, side by side in the same grasslands. And this is an example here in northern Argentina. A little bit of biology for the group. We know that the males sing different songs, and this is likely a learned trait. There's possibly a genetic component to learning this, but we think they can learn different songs. They show very little morphological variation, which we think reflects that they exploit similar resources, similar grass seeds. We can generate viable hybrids in aviaries, and we think we can ID them in the field. That this is a little bit complicated for some logistical reasons. And we do know that they mate, when we looked, that they mate assortatively in symmetry. At least the one species pair where we've done the work. And so again, this is a similar experiment to the one I showed you in the Monarcha system. When we simulate these territorial intrusions, we see that we get the strongest aggressive response from males when we combine conspecific plumage and conspecific song. And so again, in this system, we think that these traits are involved in species recognition. At least we can test it with males. In terms of the genetic basis of these traits, we've done this in different ways. The one I'm showing you here is a genome in a genome-wide association study. The average divergence between the species is relatively low. The average FST genome-wide is about 0.08. And so we can do these these GWAS, in which what we used in this case as a phenotype is the pigment type and concentration in different body patches. So we've extracted the pigments and measured their concentration. We use that as a phenotype, and here we compare, for example, the throat patch from a species that has a cinnamon throat and a species that has a black throat. And we can see two clear peaks on known coloration genes. In this case, these peaks only include a single gene. If we compare, well, a single gene in this case, these are two that are close together, but both of them are thought to be involved in coloration. If we compare a species with a cinnamon throat to a species with a white throat, we find a single peak and a different one here, including another gene involved in melanogenesis. And we can do this again and again across all different plumage patches. If we do that, we repeatedly find these 6 regions that are associated to changes in pigment concentration across the different body parts. And all of them contain melanogenesis genes. So for example, these are the 3 that I mentioned before. We also have the agouti signaling protein in this case too. And that's on scaffold 252. If you look across plumage patches, you can see that peak popping up again and again. And so now I wanted to look in more detail into this peak. And what I'm showing here is a windowed FST in the same region where I've overlaid pairwise comparisons among different pairs of species. And we see that this peak is present actually in 9 of the 10 different comparisons we did in this paper. And so we can zoom into this region and build a PCA with the variants that are just under that peak. And in that case, we see that there's likely maybe 3 or 4 different haplotypes across these 5 different species. For example, the purple species here and the red one seem to share the same variants in this region. Now I've added this third panel on the right, zoomed in and showing FST values for individual SNPs. And the reason I did this is to show you that around the agouti signaling protein, most of our differentiation, well, actually all of our differentiation is non-coding. And so we think what are involved here are different regulatory regions. We also, as an aside, used an earlier version of the pipeline I explained from Monarca, in which we were able to show that these regions have likely undergone different selective sweeps. And so the last thing I wanted to do here is take these different, the 6 different regions and ask, do we mostly observe species-specific clades if we look at topologies from these areas, or do we see variants that are shared across different pairs or groups of species? And how does this change across the different areas? So if we see a lot of sharing, do the species change with which different species share variants at these regions? And so here I pulled out as an example a region upstream of the TERP1 gene, which was involved in one of those peaks. And you can see that, yes, we observed for this species a species-specific clade. We also can see a clade that's shared between the only 2 species that have, or the 2 species that have black throats. And then we also see sharing of variants across all the other species in the group. Here's another example in which we see the same pattern, a species-specific clade for one species, sharing between these 3 species of this clade, and then sharing of older variants in other regions. And the result is the same across the 4 remaining regions where we see sharing of newer variants, sharing of old variants, and very few species-specific clades. And so just to summarize that part, as I said before, we see both old and new shared mutations that contribute to plumage diversity. And the combination of the variant, we think that it's the combination of the variants across these different coloration genes that can lead to species-specific plumage patterns. We do see species-specific mutations. These are less common, and we only observe them in these 2 species that actually have like smaller ranges and more towards the periphery. This seems to be the exception. And so with my objective of showing these 2 examples, and also I wanted to put these into context with other examples of sort of recent bird species that differ primarily in their coloration patterns, and for which we have a good understanding of the loci that are perhaps behind these coloration differences. What I think is happening here is that the evolution of these differences in sexual signals leads to this prezygotic isolation. And this can sort of buy time for other reproductive barriers to accumulate. And so I think the larger question is, well, will these incipient species last? And I kind of like this quote from this paper by Rosenblum from 2012, where they propose this sort of ephemeral speciation model in which they suggest speciation can possibly be very common and rapid, but that these new species may not always persist. And so I think these two examples I showed today show a mechanism by which we're generating these novel phenotypes that matter to reproductive isolation. The birds care about them. What happens next, I'm not sure. I think some of these cases may merge eventually back in to other species, and others may not. And geography may have something to do with that too. And I wanted to end, there's a lot of people to think. I wanted to just leave that up there, and maybe we can move to the discussion.
Thank you for a great talk about two fascinating systems.